Hello everyone. In this video, we will talk about polymerase chain reaction. In simple words, polymerase chain reaction or PCR is a technique used to make several copies of small fragment of DNA or RNA. If you understand the meaning of the word in PCR, you can better tell the process from its name itself. PCR is made up of two words, polymerase and chain reaction. Polymerase means an enzyme that makes polymer of any other molecule, which in this technique is DNA. And chain reaction is a type of chemical reaction which progresses in an exponential way. Or in simple terms, we can understand that if a reaction produces two molecules, the second reaction makes 4 and the third reaction makes 16 copies. Like that, 32, 64, 128 and 5, 112 and so on. So, in a matter of just few hundred reactions, we can produce billion of copies of DNA. The next question would come to our mind is that why to make billion copies of DNA? What are its uses and purpose? The reason for this is only after making billions of copies, we could efficiently detect the presence of target. For example, if you have only 10 copies of bacteria genome, without amplifying to billions of copies, you were not able to detect it. PCR has thousands of uses like diagnosing infections and infectious disease. For example, it is used to diagnose infections by virus. If we take a blood of a patient that has been infected by a virus, then we will amplify the DNA of the virus so that we are able to study which type of virus it is and its property. Similarly, PCR is used in hundreds of fields like crime investigation, genetic research and molecular biology, etc. Now, let's first talk about the things we need to perform PCR and then we'll discuss how this process happens. The most commonly used equipment in PCR is the thermal cycler, also known as the PCR machine. Inside the PCR machine, we have these small tubes in which all the chemicals are inserted and reaction takes place. Now, let us look at the stuff that we need to put inside these tubes that make this reaction possible. The key ingredients of PCR reactions are tag polymerase, primers, DNA template, nucleotides. Now, let us learn about them in details. Tag polymerase. It is a type of DNA polymerase. So, in PCR, it requires a DNA polymerase enzyme that names new strand of DNA using existing strand as template. The DNA polymerase typically used in PCR is called tag polymerase, which is isolated from heat-tolerant bacteria named Thermus aequitas. This bacteria lives in hot spring and its DNA polymerase is very heat stable. The second important thing we need to perform PCR are the primers. DNA polymerase need primers to start the reaction, as the polymerase cannot initiate its reaction but can only propagate it. PCR provide a short sequence of nucleotide, usually around 20 nucleotides in length. Primers provide a starting point for DNA synthesis, as we know, and primers are also important as they help to select the exact position of DNA that will be amplified. And we use two such primers in each PCR reaction, which are designed so that they isolate the target region of the DNA to be copied. Coming to the next one is the DNA template. It is nothing but the segment of original DNA which we want to amplify. And nucleotides, as we know, these are the basic building blocks which are used for DNA synthesis. Coming to next one is the process of PCR. The PCR process has 4 steps: collection, preparation, amplification and post-PCR cleanup. The PCR machine step happens in the amplification step. It begins with a segment of DNA sample placed in a suitable tube along with the reagent and chemicals listed above. The tube is placed in a PCR machine or thermal cycler. The thermal cycler takes the solution to a 3-step process: denaturation, annealing and extinction. Step 1. Denaturation. The solution contained in the tube is heated to at least 94 degrees centigrade using a thermal cycler. The heat breaks the hydrogen bonds of the original DNA sample and separates the DNA into single stranded. This is termed as denaturation of double-stranded DNA. Step 2. Annealing. The sample mixture is then cooled to between 50 to 60 degrees centigrade, allowing the DNA primers and the DNA polymerase enzyme to bind to the individual strands of DNA that were separated by the heating. This is termed annealing of the primers. At this point, the nucleotides A, T, G, C from the added mixture solution will pair with the individual separated strand of DNA that results from the heating process. Step 3. Extinction. Once joined together, they form a new complementary strand of DNA, termed as extinction of the DNA. Thus, a new duplicate double-stranded DNA molecule has been formed from each of the single-stranded of the original sample molecule. The temperature cycle from 95 to 50 to 60 degrees centigrade. The cycle is then repeated about 35 to 40 times using the thermal cycler, which automatically repeats the heating and cooling cycles of the process. Resulting DNA sequence is doubled each time the heating or cooling cycle is conducted by the cycler. Thus, what started out as a single short segment of DNA from one sample can be amplified to form millions of copies after 35 doubling cycles. Step 4. Analyzing with electrophoresis. Once the PCR process is completed, the resulting amplified segments generated can then be compared to other nucleotide segments from a known source. The PCR generated nucleotide sequence are then placed next to the known nucleotide sequence from humans, pathogens or other sources in a separating gel. Electric current is then run to the gel and the various nucleotide sequence within the gel form bands that resembles a ladder according to their electric charge and molecular size. This is termed as gel electrophoresis. Bands or ladder-like steps that migrate to the same level in the gel show identity of nucleotide sequence. This is one of the most popular way PCR tests are completed. Coming to next one is PCR cloning method. PCR cloning differs from traditional cloning in that the DNA fragment of interest and even the vector can be amplified by polymerase chain reaction and ligated together without the use of restriction enzyme. PCR cloning is a rapid method of cloning genes. It allows for the cloning of DNA fragments that are not available in large amount. Typically, a PCR reaction is performed to amplify the sequence of interest and then it is joined to the vector via a blend or a single based overhang ligated prior to the transformation. Earlier, PCR cloning often used tag DNA polymerase to amplify the gene. This results in a PCR product with a single template independent base addition of an adenine residue to the third prime end of the PCR product through the normal action of the polymerase. These A-tailed products are then ligated to a complementary T-tailed vector using T for DNA ligase followed by transformation. DNA polymerase are now routinely used to amplify the sequence with the PCR products containing 3' extensions. The blantain fragments are joined to a plasmid vector through a typically ligated reaction or by the action of an activated vector that contains a covalently attached enzyme, typically topoisomers I, which facilitates the vector's insert joining. Some PCR cloning systems contain engineered suicide vectors that include a toxic gene into which the PCR product must be successfully ligated to allow propagation of the strain that takes up the recombinant molecule during the transformation. Coming to advantages, high efficiency with dedicated vectors, enable to high throughput. Disadvantage, limited vector choices, higher cost, lack of sequence control junctions, directional cloning is difficult, multi-fragment cloning is not straightforward. That's all about PCR.